Okay, so I'm going to go over the practice genetics problems part 2. You should have your sheet out in front of you so you can kind of check your work. Remember, you want to attempt these on your own first and then use this video really to check the work that you've done. So let's start with question number 1. If the offspring produced from the mating of 2 pink flowered plants, okay, so we got 2 pink flowered plants, contains 1 red flower producing plant, 1 white, and 2 pink. So look, there's a 1 to 2 to 1 ratio. 1 red to 2 pink to 1 white. We started off with 2 pink flowers. Okay? What would be the simplest way of explaining the inheritance of flower color in these plants? That should bring you back to the 4 o'clock flowers and the snapdragons. And so this is a great example of incomplete dominance. That would be the easiest explanation here. So again, how did I know that? Well, I saw a 1 to 2 to 1 ratio, and the 2 here were pink that looked like the parents. Okay, so how would we write that? So again, you should put incomplete dominance. That's really the answer to the question. Okay, and then this is show your work, so you should really do a Punnett square here. So for our pink flowers, remember we're going to use R1, R2. There's 1 pink parent. We're going to mate that parent with the other, R1, R2. So those are both pink flowered plants. And so if I were to set up my Punnett square, I'm going to put this parent on the left. So R1 and R2. So again, 50% of the gametes contain R1 and 50% of the gametes contain the R2 allele. And the same thing across the top for the other parent. Okay, we're showing how those alleles were split up into gametes. And then inside my box, I'm going to put potential offspring. So my first here is going to be R1, R1. Hopefully you've gotten pretty good at these Punnett squares by now. My second is going to be R1, R2. Here I've got R1, R2, and then R2, R2. And so this R1 here is going to be my red flower producing plant. These 2 heterozygotes, so R1, R2, are going to be my pink flower producing plants, and this last square here, R2, R2, is going to be my 1 white flowered plant. Okay, so again, that was number 1, and the answer here is incomplete dominance, and again, your Punnett square is your proof. Let's go to number 2. So number 2 is an example of multiple alleles. Okay, it says in rabbits there are 4 alleles. So anytime you have more than 2 alleles, it's an example of multiple alleles. So you've got 4 alleles that exist for coat color. You've got capital letter C, brown. CCH gives you the chinchilla. CH gives you the Himalayan color. And lowercase c gives you an albino rabbit. It says the alleles are listed in their dominance hierarchy. So brown is dominant to chinchilla. Chinchilla is dominant to Himalayan, and Himalayan is dominant to albino. What would the offspring of a cross between a homozygous chinchilla rabbit, okay, so we've got CCH, CCH, that's a homozygous chinchilla rabbit, and a brown rabbit with the genotype capital C, lowercase c, so crossed with C, brown rabbit. So let's go ahead and do our Punnett square. So I'm going to put this parent on the side. Now this parent only has the CCH allele, so I can simplify my Punnett square and just write this once, because 100% of that organism's gametes will have the allele CCH. Now across the top I have to do a little bit more work, right, because we have 2 different alleles. So half of the gametes will contain the big C, and half will contain the little c. Okay, so now inside my box here, my first one is going to be, I'm going to put the capital letter C before the CCH because we know that brown is dominant to chinchilla. So that brown allele, capital C, is dominant to the CCH allele. That would be a brown rabbit. Okay, now over here we know that CCH is dominant to lowercase c, right? Chinchilla is dominant to albino. So I'm going to put the CCH first, right? But whatever the dominant allele is, I'm going to put it first. And then C. Okay, so the answer here would be a 1-to-1 phenotypic ratio. 1 brown to 1 chinchilla rabbit. Okay, let's erase that one. Move on to number 3. So number 3 says, if a man with type AB blood, okay, so let's start with that. So we have a man here, and he is AB. So let's think about what kinds of genotypes produce the AB phenotype. Remember, this is an example here of codominance. Both the A and B surface carbohydrates are produced. Okay, so the genotype IA, IB is the only genotype that can give us that phenotype. Okay, so this man has both the IA allele and the IB allele. Okay. And he mates with a woman that's type O. She's type O. So how do we get type O? Hopefully you remember that the only way to get type O is if 2 lowercase i's are inherited. Remember, blood type is also an example of multiple alleles because there's 3 different alleles. So there's different potential combinations of those. But the only way to get type O blood is little I, little I. Okay, then it says, what percentage of their children will be type B? Okay, so let's cross these guys. Here's my Punnett square. So I'm going to put the woman on the left. Okay, so there she is, little I. That's all she has, so all of her gametes are little I. And then I'm going to put my man up at the top here. So half of his gametes are IA and half are IB. And so remember, inside the box, we're going to put potential offspring. So the first one, we're going to put IA, little I. Notice the order that I put them in, and that's because the IA allele is going to be dominant to the little I. Okay? And then in my second box here, IB, little I. And so it says how many are type B, and you would say 50%, right? Because it's a 1-to-1 ratio, or half would be the IB, little I, or blood type B. Okay, so that's number 3. Number 4, let me get this erased. Number 4 says, both Mrs. Brindisi and Mrs. Crocker had baby girls the same day in the same hospital. Mrs. Brindisi took home a baby girl who she named Lily. Mrs. Crocker took home a baby girl that she named Maddie. Okay, so let's keep things straight here. So I'm going to put Mrs. B, Mrs. C, Mr. B, Mr. Crocker. Okay. The Brindisis have a baby girl named Lily, and the Crockers have a baby girl named Maddie. Okay. It says, Mrs. Brindisi's baby was very crabby, so she began to suspect, or maybe hope, that her child had been accidentally switched with the Crocker baby in the hospital nursery. Blood tests were made, and the results showed, okay, so Mrs. Brindisi is type A. No, hold on. I got that wrong. Okay. Mr. Brindisi is type A. Mrs. Brindisi is type B. Okay. Mr. Crocker is type A. And Mrs. Crocker is type A. Okay. Lily is AB. And Maddie is type O. And the question is, had a mix-up occurred? And then you're going to show Punnett squares for proof. Okay, so you have to know how each of these blood types is formed, so what the genetics is behind it. So for type B blood, Mrs. Brindisi could have been IB, IB, or IB, little I. And Mrs. Brindisi, or sorry, Mr. Brindisi type A blood could be IA. Let me actually write this. So for type B, we could have IB, IB, or oops, sorry, hold on. Much easier writing on a board. IB, little I. Both of those is going to give you type B. Type A is going to be IA, IA, or IA, little I. And those hold true for both Mrs. Crocker and Mr. Crocker, who are both type A. So this would be their potential as well. Now Lily has to be IA, IB. You could do a capital I, or I'm sorry that I wrote it like that. And Maddie's going to be little I, little I. Okay. Okay, so the question is does Lily belong to Mr. and Mrs. Brindisi, and does Maddie belong to Mr. and Mrs. Crocker? And hopefully by looking at this, you'll say, yes, a switch has not occurred, right? So there's been no mix-up. Mr. and Mrs. Brindisi are opposite blood types, right? Mr. is A and Mrs. is B. And so, yes, it's definitely possible for them to have an IA-IB child. Okay? And you could do all of the different Punnett squares that could give you this outcome. Okay? So, any of these genotypes that we listed could produce the IA-IB genotype in their child. Now, Mr. and Mrs. Crocker, to have a type O child, both of them would have had to have been IA, little I. So when you go to set up your Punnett square, you're going to have to choose that genotype. Okay, and again, you can choose either of the genotypes for Mr. and Mrs. Brindisi. Both of those are capable of giving you an AB child. Okay, let's go on to number 5. So let's erase this. Okay. So number 5 says, in humans, hemophilia is a blood clotting disorder. So in humans, hemophilia is inherited as a sex-linked recessive trait. Okay? So this is really important. So it's sex-linked, so you have to make sure that you write it that way. Okay, so let's see. So a woman who has hemophilia would have to be X little h, X little h. She would have to get 2 doses of the recessive allele in order to have hemophilia, where a man would be X little hy. So both of those genotypes is going to give you hemophilia. Okay? All right. So it says, suppose a normal woman, okay, so let's box that off. We have a normal woman, so she's going to be X, big H, X, we don't know. That second allele could be a big H or a little h. Both of those would give you a normal phenotype. Okay, that's what we know so far. So a normal woman mates with a normal man. Okay, so we got a cross here, and the normal man would be X, big H, Y. Okay, does not have hemophilia. So they mate, and they have 4 normal children. So they have 2 boys. Those boys must both be X, big H, Y. That's the only way to get normal in boys. And they have 2 girls. And both of them, we don't know. They could be big H, or big H, little h. So we're going to put a dash on that second X. Okay. Then it says, but one of the girls, so one of these girls, has a son that has hemophilia. Okay, so son. That is X little h, Y. Okay. So it says, what is the genotype of the man and woman, the original parents? And then what are the genotypes of their children? What is the genotype of their son who has hemophilia? And did the son inherit this condition from his mother or father? Okay. So first off, one of these girls is going to be X, big H, X, little h. Okay, because in order to have a son, mom always gives the X chromosome in boys. Okay, the Y comes from dad, the X comes from mom. So if the son has that recessive little h allele, he must have gotten that from his mom. And so one of the girls, the mom of this son, must be a heterozygote, a carrier of that recessive nasty allele. Okay? So she would be X big H, X little h. We don't have enough information to tell us what the other girl is. Okay? So we would say one is definitely X big H, X little h. The other we're going to keep as X big H, X dash, because we're not sure. Do we know anything about these original parents? Well, we know dad, right? But how about mom? Again, in order for mom to have a girl that has this genotype, mom must also be X big H, X little h. Okay? Because that recessive little h allele had to come from somewhere. It didn't come from dad, right? Because he's normal and he only has one X chromosome, so it must have come from mom. So you can figure out the genotype of everybody in this problem except one of the girls. One of the girls is going to be unknown still. Okay? All right, let's do another one. So that was number 5. Oh, where's my eraser? Okay, let's go on to number 6. So number 6 says, fur color in cats is determined by a sex-linked gene. So again, you're going to write it the same way. It's X-linked, so you have to do X with a superscript to the right, showing that allele. It says, the genotypes Big B produce black fur color. Okay, so X, Big B, X, Big B equals black. Okay, and that means also that X, Big B, Y equals black. Okay, so that Big B allele gives you the black phenotype. And then it says little b produces yellow. So X little b, or X little by, these are going to give you yellow. And the heterozygous condition, so X big B, X little b, gives you what's called the tortoiseshell. Okay, tortoiseshell. Now you can't have X big B, Y little b. The Y chromosome doesn't carry this gene. And so there are no tortoiseshell males. Okay, it's actually a little bit more complicated than this, but we'll keep it simple. Okay, so let's get to the problem. So it says, what would the result of a mating between a tortoiseshell female and yellow male be? Okay, so a tortoiseshell female is going to be X big B, X little b. And you have to write it like that because remember it's X-linked. Crossed with a yellow male. So this is going to be X little b, Y. So let's do our Punnett square. I'm going to put mom on the side. So she's going to be X big B, and half of her gametes are going to be X little b. And we'll put dad on the top, so he's got an X little b and a Y chromosome in his other gamete. Okay, so the X and Y chromosome in a male are homologous and they sort during meiosis. Okay, so inside we're going to have X big B, X little b, so that's a tortoiseshell female. X little b, that's a yellow female. X big B, Y, there's a brown male. And X little b, Y. So your answer should be half of the females are tortoiseshell and half are yellow. Half of the males are black and the other half are yellow. Okay? Okay, let's get this erased. So number 7 says, in leghorn chickens, okay, colored feathers are due to the dominant gene, capital C. Okay, so capital C equals colored feathers. Okay, while feathers, the white feathers are due to the recessive allele. Okay, so little c equals white. Feathers. It says another dominant gene, capital I, inhibits the expression of color in birds with genotypes big C, or big C, little c. Okay, so this is a case of epistasis. Remember that? In lab, we went over an example with, I think, mice, or sorry, in lecture, an example of mice and the Labrador retrievers. Both of those were examples of epistasis, dealing with color as well. And in lab, it was the corn cob. It was that 13:3 ratio. Actually, you'll be working on that lab this week, so you may not have done that yet. Okay, in both of those cases, and in this case as well, we have one gene controlling the expression of another. So the I gene controls the expression of the C gene. Okay, so let's put inhibits color. And so little I allows color, right? Okay. Okay, so it says this is an example of epistasis, and consequently both, okay, so dash, dash, I dash, and little c, dash, dash, give you white feathers. Okay, so what does this mean? This means that if the capital I allele, which is the inhibitor allele, is present, it doesn't matter what's there for the color gene. It can be big C, big C. The chicken will not have colored feathers. The feathers will be white. And the same thing is true for that little C allele. If you get 2 doses of the little C allele, doesn't matter what's there for the I gene, the feathers will still be white. Okay? So let's get to the actual question here. So it says, suppose a colored male chicken. Okay, this is not sex-linked. Nowhere did it say sex-linked, so don't start putting the X chromosome. It's a colored male chicken that is homozygous for the color gene. Okay, so big C, big Z, C, homozygous for the color gene, and he's colored. So that means he must have 2 doses of that little I allele, because if the big I was present, there would be no color. We're going to mate him with a white hen that is heterozygous for both the color gene and the inhibitor gene. So big C, big I, little I, heterozygous. Show a Punnett square for this cross, and then what phenotypes would be produced and in what ratio. Okay, so let's start off with this parent. This parent has 2 of the same alleles for both genes, and so it's really easy here. All of the gametes of this parent are going to be capital C, lowercase I. That was easy. Okay, now the second parent is different. In the second parent, we've got to do this, right? So 1/4 of the gametes will be big C, big I. And then this, 1/4 of the gametes are going to be big C, little I. And then this, so 1/4 little c, big I. And then 1/4 little c, little I. Okay. Okay, so now we're going to put potential offspring in the inside. So big C, big I, little I. Okay, this will be a white chicken, right? Because, I'm going to put a W underneath it for white. Because, yeah, so it gets 2 doses of the color gene, but since that inhibitor allele is present, we're not going to have any color. So it's going to be a white chicken. Second chicken, big C, big I, or sorry, little I. Okay, so the inhibitor gene is the allele for inhibition is not there, right? We have that little I allele that allows color. And we've got 2 capital letter C, so that's definitely going to be a colored chicken. And here we've got big C, big I, little I. Okay, as soon as we see that big I, we're going to go with white. And then this is supposed to be a line over this. Okay, big C, little I. Okay, so that's going to be a colored chicken. So we have 2 coloreds and 2 whites, so we're going to do a 1-to-1 ratio of colored to white. Okay, that's it. So let me get this erased. We'll go to the next problem. Okay, so number 8 says in rabbits, a dominant gene produces spotted body color. And its recessive allele, solid body color. Another dominant gene produces short hair, and its recessive allele, long hair. Okay, so let's get this straightened out here. So we have spotted and solid. So, and spotted is dominant, correct? Yeah. So we've got spotted equals a big S. Solid, we'll make it a little s. So there's one gene. The second gene has to do with hair. We already used the letter S, so let's use the letter H to represent hair. So short, I believe, is dominant. So we got short hair. And then we've got long hair is recessive. Okay? It says rabbits, heterozygous for both characters. Okay, so big S, little s, big H, little h. So that's a heterozygous rabbit, spotted with short hair, is going to be mated with homozygous recessive rabbits. So little s, little s, little h, little h. So solid with long hair. The results of the cross were as followed. Spotted with short hair. I want to write these out. Spotted with short hair, we have 96 rabbits. Solid short, we have 14. Spotted long, we have 10. And solid long, we have 80. Okay, so let's look at these numbers. There's not an equal amount of each of them, right? From this particular cross here, a heterozygote with a homozygous recessive dihybrid, if the genes were not linked, we would expect equal numbers of offspring here. We would expect a 1-to-1 to 1-to-1 ratio. We don't get that. We get a whole lot more spotted short and solid long than we do solid short and spotted long. Okay, we get 96 and 80 compared to 14 and 10. So it's a good indicator that these genes are linked. Let's take a closer look. Spotted short, the first one, are either of the parents spotted short? Yes. This parent is spotted short. So that's a parental phenotype. And then the same thing here. With solid long, we got our 80 here that are also solid long. So these two are parental. They look like one of the two parents. Where these two here, solid short and spotted long, these are recombinants, right? They don't look like either parent. How were they formed? Must have been through a recombination event, okay, that happened in this parent. Okay, so you gotta take yourself back to meiosis. And the S and H gene, again, are on the same chromosome. And so in that parent, the capital S and the capital H, right, coding for spotted and short, they're on the same chromosome. And the other two alleles, little s, little h, giving you solid long, are also on the same chromosome. So these two make sense. Okay, however, how do we get these? Well, there had to be a swap of either a big S and a little s, or a big H and a little h. Okay, there must have been a swap. So again, you got to take yourself back to meiosis 1. During prophase 1, when homologous chromosomes are in tetrads, and there's the potential for a crossover or a recombination event, that must have happened here. So the question says, what evidence for linkage is shown in the cross? And you would do just this, the numbers here. Okay? If there wasn't linkage, again, you would expect an equal number of each of these phenotypes to be produced. It says show a Punnett square. Okay, so let's do that. Okay, so in our Punnett square here we've got, okay, so one parent that is big S, big H, little h crossed with little s, little h, little h. Let's put, oop, that's not a B, it's an H. Let's put this parent on the left-hand side. So little s, little h. And we'll put the other parent at the top. So big S, big H, big S, little h, little s, big H, and little s, little h. Okay, here's my recombinant gametes right there. So in my first box, I got big S, big H, little h. Okay, in my second box, I've got big S, little h, little h, little s, big H, little h, and then little s, little h, little h. And if we look at our numbers that were produced, again, if they weren't linked, you would expect a 1 to 1 to 1 to 1 ratio. But we don't get that, right? We get 96 here. We get 14 here, 10 here, and 80 here. So again, these are my recombinants. Okay? We could figure out that ratio. And to do that, we would take the lowest number there and make it our 1. Okay, so what does that mean? That means that our 10 here, 10 divided by 10 will be 1. And now we're going to divide everything else by 10. So 96 divided by 10 is 9.6. Okay, so we have a 9.6 to, let's see, 14 divided by 10, 1.4. So a 9.6 to 1.4 to 1 to 8, right? 80 divided by 10 is going to give you 8. So that's our phenotypic ratio, 9.6 to 1.4 to 1 to 8. Okay, you have to do those ratios in lab too, so hopefully that helps you. The question didn't ask for that ratio, but provided it just so that you can see it. So we didn't get a 1 to 1 to 1 to 1 ratio. We got something very different. We got a whole lot more of our 2 parentals and a whole lot less of our 2 recombinants. So again, that's evidence that the genes are linked. And so the next part of the question says, calculate the recombination frequency and the map distance between the 2 genes. Okay, so how do we do this? So to figure out recombination frequency, we take the number of recombinants and divide that by the total number of offspring. So that would be the number of recombinants is 14 plus 10 divided by everybody. So 14 plus 10 plus 96 plus 80. Okay, so let's do that first. So this would be 24 will be our numerator, and our denominator here is going to be 200. And so that's going to give us 0.12. We need to multiply that by 100 to get a percentage. So it's going to be 12%. So the recombination frequency between the S and H genes is 12%. They recombine 12% of the time. The distance, remember 1% recombination equals 1 map unit, so we would say that they are 12 map units apart. Okay, and that's it for that one. Okay, so number 9. Okay, so we're on to number 9. Number 9 says, in Drosophila melanogaster, so the fruit fly, the genes for wing length and body color are known to be about 20 map units apart. Okay, so automatically you know that these genes are linked. So there you go. So they are linked and they are 20 map units apart. Okay, that's important. So it says alleles for normal wing length long and gray body are dominant to alleles for vestigial short wings and black bodies. Okay, so we're going to say normal wings equals, we'll use big V, where vestigial wings is recessive, we'll use a little v. And then our second gene here is color, and we said that gray is dominant to black. Okay, so gray is dominant to black. Right? So black. And remember, you can pick any letters to represent these alleles. That's fine. Okay. And we know that they're linked. So the wing length and the color gene are on the same chromosome. They're going to be inherited together. Okay. So it says next, flies homozygous dominant for these genes. Okay, so big V, big V, big G, big G. So these would be normal winged gray flies are mated with homozygous recessive flies. So little v, little v, little g, little g. Okay, so those would be vestigial black. Okay, these are the parents. The result of that, our F1 generation, are going to be all flies that are heterozygous. So big V, big G, little g. Okay, so those would be normal gray. We then test cross those. Okay, so as soon as you see that word test cross, you should say that's a homozygous recessive fly. Okay, whenever you see test cross. If there were 100 offspring from this cross, how many would you predict would be the recombinant phenotype? Okay. So you can go ahead and you can do the Punnett squares here, but look, it tells you that these are 20 map units apart. So the recombination frequency is 20%. And so that means 20% of the offspring will be recombinant. If you have 100 offspring, that means 20 of them will be recombinant flies. Okay, 10 will probably be vestigial gray, and 10 will be normal black. Okay, or 10%. Okay, will be of each. You can go ahead and you can do the Punnett square though, just for some evidence. Okay, and again, if you had 100 offspring, 20 of them should be of those two phenotypes, right? So vestigial gray and normal black. And the other 80% should be the parental, normal gray and vestigial black. Okay, let's do that Punnett square. So I'm going to erase this slide. So again, we've got big V, big G, little g. And we're going to cross that, test cross it. So little v, little g, little g. Let's put this parent on the side because it's a little easier. Little v, little g. Okay, and this parent we're going to put across the top. So again, the combinations here will be big V, big G. That's a parental. Little v, sorry, hold on. I'm jumping ahead of myself here. Okay, so big V, little g. Quarter of the gametes are going to be little v. Actually, no. Some of the gametes here will be little v, big G. And then little v, little g. Okay, so these are recombinant gametes. These are parental. You got to go back to the parents of this guy. The parents there were homozygous dominant and homozygous recessive. And so that means the two dominant forms of this gene, big V, big G, are on the same chromosome, and the recessive forms of each gene, little v, little g, are on the same chromosome. So they're going to not sort independently, right? They don't follow Mendel's second rule. So let's go ahead and let's fill in our box. Let me grab a different color pen here. And I'm outside on my back porch, so I'm sorry about the dog and the mowing. So here we go, big V, big G, little g. So that's going to be a parental, normal winged gray-bodied fly. So I'm going to put a P under here for parental. And then my next box here is going to be big V, little g, little g. And this is going to be a recombinant. Okay. And it's going to be a vestigial black fly. Doesn't look like either parent here, right? Either one of these are vestigial black. My next offspring is going to be little v, big G, little g. That's a recombinant. So this is going to be a vestigial gray fly. Doesn't look like either parent. And then my last here is going to be little v, little g. And this looks like that second parent up there. So it's parental. It's going to be a vestigial black fly. Okay, so these right here are recombinant, and if we have 100 total offspring, and these genes are 20 map units apart, they recombine 20% of the time. 20% of 100 is 20. So 20 of the flies should be of these two. Okay, probably 10 of these about, and about 10 of these. That means the other 80, so about 40 and 40 of those offspring, roughly, should be of the parental phenotype. Okay? One more to go. The last one has to do with mapping. Okay? And so I'm going to start off with a straight line, and that represents my linear chromosome. And now I'm just going to place the genes in the order that's kind of provided here in this question. Okay, so it says the crossover frequency between linked genes A and C is 40%. So I'm just going to create a spot here. That's going to be my A gene, and here this is going to be my C gene, and I know that these guys are 40 map units apart. Okay, we got a couple of other genes to put on here, and we're trying to figure out the linear arrangement. And so the next part of this says, okay, so between the B and C gene, it's 20. All right. So am I putting it in the middle of A and C or on the other side of C? Let's see. I need to figure out where is B in relation to A. So I got to skip over a little bit. So between A and B, it's 20%. So 20% between B and C and 20% between A and B. It's going to go right here. So there's the B gene because this is 20 and this is 20, equaling a total of 40. One more gene to put on here, the D gene. Okay, so between C and D, it's 10. Between B and D, it's 10. Okay, so right there, it needs to be here. Oop, sorry. Okay, so between B and D is 10, and between D and C is 10. And we even give you between A and D is 30. So here, yeah, 20 plus 10 is 30. So that works out. So the linear order, or the sequence, is A, B, D, C. Okay? And that's the last one. So hopefully you understood these. If not, don't forget, you can always come to office hours. You can rewind this and watch it again. Hopefully it helps you. So again, you should check all of your work on your worksheet. Make sure that you got the same answers as me before you go ahead and turn that in.